So I'm talking about a new approach, a technology approach, which helps us to isolate novel bacterial strains from human origin. And we are a company which is based in Paris. We are working off a technology platform which uses tiny droplets, picoliter scale droplets on a lab-on-a-chip platform, which helps us to culture and screen at the same time bacterial populations. Obviously, we can do this in extreme throughput, as we can have about more than 30 million bacterial populations in parallel, which we can culture and screen. The technology which we are using is based on technology licensed in from Harvard University, from the Broad Institute in the US, from ESPCI in Paris, and of course also technology and patents which we have filed on ourselves. We use that technology to develop novel consumer health products, mainly based on the dysbiosis or on addressing lifestyle diseases which are caused by dysbiosis, and where we want to provide novel products to consumer health to restore a robust and resilient microbiota. But of course, as all of you know, many of those bacterial or beneficial bacteria which populate the human body or are living inside our human body are very difficult to isolate, and the more difficult to isolate them on specific traits. And that is known in microbiology as the Great Plate Count Anomaly, meaning that if you take an environmental sample and look on one side by classical methods like microscopy, nowadays probably by metagenomic analysis, on the diversity of the bacterial species in that sample, you observe a much higher diversity than what you can culture as bacterial species in the laboratory. And there are potential reasons for this and many hypotheses around that phenomenon. One is that the growth state in nature, such as dormancy, is not going to wake up in the laboratory conditions, that there are specific growth conditions or growth factors missing which are difficult to replicate under laboratory conditions, and that there are of course also a lot of interactions between bacterial species and other bacteria or other organisms or with human host cells in order to drive proliferation of the bacterial cells. There are technology approaches which address that problem. One is, for example, the iChip technology, which is a modular device which allows to culture bacterial cells in a kind of a chip-like structure, and it's tried to provide the correct environment by inserting the microbial sample in such a device into into the appropriate environment where it's supposed to have the growth conditions and growth factors being available. But obviously that cannot be used in any case for any type of application or biological sample where you would like to apply that. Another solution is dilution to extinction, so meaning diluting the microbial sample onto a single cell level, replicating this in high throughput in microtiter plates, but of course also kind of a limited approach and also not really a high throughput approach. So the challenge is really how can we deconstruct the microbiota in order to harness novel strains for product development in the consumer health business, where we address dysbiosis, which is the cause of many lifestyle diseases, as we have heard earlier today, like obesity, like diabetes, like allergy. And how do those strains which are isolated modulate the microbiota? And effectively, what one would like to do is to provide those strains as a supplement or as a drug to prevent such diseases. Obviously, what you need to do is to isolate live strains from human microbial samples, and in order to work with them, on the other side, of course, also to understand the mode of action and how they restore a robust microbial community on the body. What we are using for that is a lab-on-a-chip technology. The lab-on-a-chip technology is based on droplets, which are generated as water and oil emulsions. And what we can do with those is that we can culture on a single cell level bacterial species and can grow them in those droplets for a prolonged time, so like 7 days or 14 days. And on that same kind of pool of bacterial populations, we can then deploy certain assays for which we can select for desired phenotypes of those bacteria. And due to this extreme throughput which we can generate, we can select something like 30 million variants or 30 million strains on a single day. That is just for illustration purpose. Of course, nobody is doing high throughput automation for such purpose for 3 years. Would biologically make no sense, would commercially make no sense. It's just for comparison how more effective droplet-based microfluidics is for solving that problem. So how is it done? How do we do that? In principle, we are replacing microtiter plate wells by water and oil emulsions. These are droplets which have a volume of about 20 picoliters, so we are driving down volumes millions-fold. And by that, we increase the throughput dramatically. We can generate those droplets at a rate of 2,000 droplets per second. And we cannot only generate those droplets, of course we can encapsulate cells or bacterial cells into the droplets. And the droplets environment is very stable, but also prone to a variety of culture conditions. So we can keep the cells in the droplets, we take them off the platform and can incubate them in a standard incubator. The droplets are gas permeable, so they acquire the atmosphere which we require, whether that is under anaerobic conditions or aerobic conditions. In principle, you can culture them in standard incubators. But it's not the only feature that we can culture and screen them. We can also manipulate the droplets. It's like lab on a chip, like a liquid handling platform with different capabilities of incubating the droplets. We can also add reagents to the droplets like enzyme substrates. We can deploy a range of different assays to the droplets, and I will cover that in a little bit later. And most importantly, what we have observed, and we'll show data on that, is that we see a lot of those bacterial species which we usually cannot easily take into culture under laboratory conditions can also grow in droplets. So we can look at a much broader diversity of microbes in droplets and at the same time deploy assays to select them for desired traits. The reaction environment is highly reproducible, so the monodispersity of the droplets is very high. Polydispersity is less than 3%. And the screening environment is very adaptable. So we can work with complex media. We have, for example, worked with complex stuff like milk. We can have different incubation conditions. Droplet microfluidics is used also in life sciences, for example, in PCI applications, which demonstrates how stable that droplet environment is. That slide depicts kind of a few of those handling capabilities on a microfluidic chip, like, for example, incubation droplets on the chip, but we can do this, of course, also offline, mixing reagents, and especially when we isolate bacteria, grow them in droplets, and have to incubate them for a prolonged time, like 7 days, 10 days, 14 days, we can simply take them off the chip, incubate them in Eppendorf tubes, so to speak, because the volume is tiny. And then we can re-inject them on the chip for further analysis. And analysis done usually using fluorescent detection. So we have a 4-color detection capability on the platform. And at high speed, we can interrogate the signal of the droplets and can sort the droplets according to the fluorescent signal which is being generated as positive or negative population. We can also set different thresholds, so we can bin them into different populations in principle. That depicts kind of a comparison of a standard microbiology workflow with droplet-based microfluidics. So we start, of course, off with the same type of sample material, sample preparation, in order to purify the microbial samples to keep the live strains. What you then observe if you select for growth on solid media is that the number of microbes goes down dramatically, as I have described before. And then in principle you hope that among those few colonies which you can grow from the original sample, that there are some colonies or some bacterial species among those to which you can deploy your assay and might become a hit candidate for the product development. So that is actually combined in a single step on the microfluidics platform, as we can isolate the bacteria or encapsulate them on a single cell level. We grow them like on agar plates, but with the distinction that we have a much higher diversity. And then at the same time, without going to microtiter plate wells, we can assay them for desired assays, like enzyme activities, binding assays, etc. And once we have selected those positive droplets or hit candidates, we can dispense them into microtiter plates as a single droplet and can grow them then actually also in microtiter plates. So that was the other very important kind of observation, that once they grow in those tiny droplets, you can dispense them into larger volumes and you can keep them growing. But again, it takes a very long time, like 7, 10, 14 days, and the kinetics or growth kinetics of the different bacterial species is quite different in those natural samples. So that depicts some of the steps. Encapsulation of cells on a single cell level, seems to be extremely important. We are currently also working with academic collaborators in order to better understand this phenomenon, which is mainly apparently based on that we create a better microenvironment for non-competitive growth. So bacterial species are not overgrown by fast growers. There's less competition for key metabolites and cofactors. There's also the kind of idea that once strains wake up out of a dormant status, that they are also able to secrete their own growth factors, which of course in large volumes become diluted and ineffective. And of course they are also devoid of any inhibitory effects of other bacterial strains or species. Then we can grow them in droplets, which takes quite a while, as I have indicated. And then we can assess those millions of microcultures on a functional basis and sort and isolate them based on that. So in summary, what this lab-on-a-chip approach with droplet-based microfluidics offers is that we can culture microbial species at ultra-high throughput, can use actually quite complex media for that. We can co-culture, what I think is also another very important trait of that technology. We can co-culture microbes with cells. We can co-culture them with reporter strains. We can directly assess interspecies interaction in droplets. And we can do that at ultra-high throughput because we can generate those 100 million droplets on the go and then automatically select for hit candidates. Also with regard to assay versatility, it offers also additional capabilities over other high throughput methods like FACS, for example, or bead-based methods, because we cannot only detect, like in FACS, intracellular markers, biomarkers, or cell surface bound markers, but we can also look for secreted biomarkers because this droplet-based environment is like a closed environment in a microtiter plate well. So that is also possible. And we can devise a broad range of assays like growth-based assays or growth inhibition-based assays. We can assess extracellular compounds, can actually run binding assays very similar to antibody ELISA-based assays, which we can run without washing in droplets by specific means. We can measure for viscosity. We can look for enzyme assays. We can look for reporter signals by co-culturing strains with reporter strains. And what is very important, as every workflow looks differently, we have, of course, the freedom to customize that workflow because there's no simple plug-and-play solution for working with different samples and assay readouts. To show you some data of the diversity which we can maintain in droplets, we also use 16S ribosomal RNA sequencing. That is where we use the kind of standard processing of trimming the sequences, demultiplexing the sequences, doing the alignment, doing the OTU clustering, and then looking at the diversity of those samples. One example which I want to present is from our scientific co-founders from Andrew Griffith's lab. In this case, they were bioprospecting for cellulolytic microorganisms out of a natural environment, in this case from a wheat stubble field. And they were comparing the diversity of microorganisms expressing cellulolytic enzymes with those using the same sample plated on solid medium, induced microorganisms for the expression of cellulolytic enzymes. And as you can see, there's a much lower diversity for the main genera on those growth-based selection on agar plates than when compared to droplet-based microfluidics. This is a workflow which we had in another collaboration project where a customer of us wanted to isolate microbes from cattle rumen, which is not that easy because, as you know, it's an anaerobe environment, so they had to apply the same kind of workflow under anaerobe conditions. So what we did was, again, processing those samples, isolating the microbes. Then we split the samples in half, directly plated them under anaerobe conditions on selective medium for growth of the microbes, encapsulated the other part of the sample in droplets, grew them for a couple of days in droplets, and then for both samples isolated DNA by 16S ribosomal RNA amplification, and then assessed the diversity by next generation sequencing. And as you can see here, again on the OTU level, we see a much higher proportion of unclassified taxa, which we obtained in droplets as live microorganisms from cattle rumen as compared to agar plates. With one of our collaborators in the agricultural area, we are looking for biofertilizers and biofungicides. And here we were looking at the diversity of genera derived from either soil microbiota or microbiota associated to plant tissue. And again, doing the very same workflow, isolating the microbial samples, splitting them in half, testing them for growth in droplets versus growth on agar plates. And what is displayed here is always the top 100 genera which were isolated from those samples as live bacteria. And the same picture holds true again, so you see a much lower diversity and lower representation of the input sample which we were using and which we were characterizing by metagenomic sequencing than what we were able to retrieve from cultured microbes in droplets in both cases. And the last example which I want to show is on skin samples, where we are working in our product development on isolating novel bacteria or bacteria which have antimicrobial activity for one very specific indication. Here we choose a slightly different approach because we have, as you might know, there are only 3 genera mainly present on the skin. Highly homologous strains, so we were rather trying to cover the complete sequence from the 16S ribosomal RNA up to the 23 ribosomal RNA, kind of spanning a 4.5 kb segment, amplifying this, running next generation sequencing analysis on it, doing the trimming and demultiplexing of the sequence data. Then we used the de novo assembly metagenome tool from the CLC workbench from Qiagen to assemble the contacts. And then we used different cutoff levels when we blasted the sequences for those different contact lengths in order to assess the diversity of samples again, where we culture microbes from skin samples on the plates as comparing this in droplets. And just as an example, I've shown here 4 different subjects. You see the same phenomenon that on plates you see less microbial strains which can be grown than in droplets. And as is not surprising, there's a big variety between different individuals. And most strikingly, for the 4th individual on that side, where we were hardly able to grow anything on the plates, and also a much lower diversity in droplets. And what it turned out for this individual is that most of the bacteria which we were obtaining in that case were Staph aureus, in principle kind of overgrowing the microbiota on the skin for that individual. For our product development, we can use 2 different approaches. We can, on one hand, use a data-driven approach, where we apply metagenomic sequencing on healthy and diseased individuals, do next-generation sequencing, assess which pathways are present in those samples, which might be interesting targets for assays with which we can isolate beneficial microbes. Then we can, based on that information, deploy assays and can run this workflow which I described before, culturing microbes directly out of their human environment and assaying them for beneficial traits. Of course, there's increasing knowledge derived from literature which we can use, and then again, it's kind of the same workflow. We identify the targets which might be interesting, can develop the respective assays, and again, deploy the assays on the screening of those microbes and isolate hit candidates for our product development pipeline or for our partners' product development pipeline. Of course, next generation sequencing and the bioinformatics pipeline is of critical importance, and there are very standard requirements which we need to have in our lab, like easy data storage and easy data retrieval. If we want to reanalyze data, we need to have robust tagging of our samples. It needs to be a very standardized workflow where we can track all different steps in the workflow, where we can also reanalyze accordingly with saved information from those workflows, and providing us with a scalable infrastructure as we accumulate more and more data. But there are, of course, also differences in different applications where we have different requirements, like for whole metagenomic sequencing, where we need to rely on high quality reference databases, which is kind of lacking today, and that is something where we are working intensely on and generating our own curated reference databases for that. Then targeted metagenomic sequences, 16S ribosomal RNA is definitely a nice tool. 16S 23 ribosomal RNA sequencing is probably a better tool, but it's still not sufficient. It's still not sufficient to differentiate many of the very kind of closely related bacterial species. Again, high quality reference databases will help there in future, but will also help for other workflow steps like chimera detection. And just a very recent paper which came out in Nature Methods, which describes a little bit this dilemma on that algorithms work extremely well if you have kind of distinct species which you want to differentiate by 16S ribosomal RNA sequence, but the closer related those strains are, the more difficult it gets with the different algorithms which are currently available on the market. The CLC workbench, which we are using and where we are also working in collaboration with Qiagen, is a very good tool for us in our laboratory for reanalysis, for getting hold on the different kind of information which are addressed with our samples, and also the trackability of the analysis workflows which we are using, so that we can also, in principle, look at the analysis history that is always saved with those workflows, so that we can reassess those workflows and have real comparisons and don't compare apples with oranges, but really oranges with oranges. And also the scalability, looking to cloud-based solutions as one other potential use, is of course for those who know about calculation time and data processing, is a very important step.
Qiagen.
Sample to insight.